Didn't touch anything? All right, cool. Hello, everyone. I would like to thank James and also Jay for also all the organizing committee for this opportunity, and I'm going to give a talk on thrombosis. So I did my PhD about 10 years ago at Georgia Tech and Emory biomedical engineering programs. So when I was at Georgia Tech, during the orientations, our department chair, Larry McIntyre, is a famous biomechanical engineer in blood flow mechanics, showed us diagrams of the anatomy of the thrombosis occurring locations. And I was very intrigued by these diagrams because it shows the thrombosis can only happen selectively to the certain areas of the vessels, and in those areas are usually bifurcations and stenosed areas. And this triggers the thinking that geometries and rheological regulations is linked to the thrombosis. And this is also my long-standing interest to understand the biomechanical regulation of thrombosis, and it's deeply implicated in natural thrombosis, and also Anna mentioned the device thrombosis. But the question is, what is the mechanism that drives this biomechanical thrombosis? It's turned out that for the last 5 years, there are lots of discoveries around this called disturbed blood flow. So disturbed blood flow is a specific type of flow patterns when the laminar flows hits obstructions, usually caused by geometrical narrowing, and then triggers the shear accelerations and then decelerations, and sometimes turbulent flows can generate it. And over the last decades, people know this disturbed flow is linked to the atherogenesis, and recently there are more and more evidence to show the disturbed flow can promote biological response. And also, the last 2 years, people harness the disturbed flow for biogenesis, stem cell differentiation. So there are lots of interesting biologies coming from the flow disturbance. During my PhD, I read these papers from Sean Jackson group in Australia. It's actually a major study demonstrate the disturbed blood flow can trigger platelet aggregations and cause the thrombosis. This is a very nice system. They did multiple systems, but I was very intrigued by this microfluidic system. They create artificial geometries that mimic the stenosis, and then they demonstrate platelet form rapid aggregations at downstream of this stenosis. And even though you inhibit this, the whole blood with collections of agonist inhibitors, and also they demonstrate this thrombosis is unprevented by aspirin or clopidogrel, which is the gold standard for antiplatelet therapies. And the most important things they link they call it biomechanical platelet aggregations with two important markers. One is the integrin alpha TB beta 3, and another one is the platelet GD1B receptors. So, and this is the set up the stage for my interest to investigate this field. And this is my work for the last 5 years to really understand the molecular mechanosensors underlying the mechanobiologies of the blood disturbed shear effect. So I look at GP1B, and recently I focus on more on integrins mediated mechanosensing. And today I'm going to mostly capture about the recent papers just accepted by Nature Materials to talk about new mechanisms from integrin alpha TB beta 3 mechanosensing. So everyone knows integrin is one of the most mechanosensors help the cells to sense the mechanical environments, and of great significance to tissue engineering, cell therapy. Fundamentally, how mechanical force coordinated with integrins exert the functions for cells is not completely understood. There are some models talk about how integrins respond to activations and how force plays a role. So the switchblade model is one of the famous one, and most of this evidence come from the crystallography. And in the last 5 years, very lucky for the field that a lot of antibodies have been available to pinpoint the different conformation of these integrins, and then allow the field to move on to apply these antibodies to look at and map integrin conformation in native environments. And so when I started my postdoc, I started to develop microfluidic thrombosis models, and then combine with the imagings, and we map the conformations states of the platelet integrins in response to mechanical stimulations versus we call it biochemical stimulation is actually indeed triggered by the agonist we put in. What we found is for the classic agonist-driven platelet aggregations, you saw very strong integrin activation in all states, whereas the biomechanical shear flow disturbance in stimulated aggregates is only positive for we call low activated integrin conformations, but not for the full activated integrin conformations. This is very interesting. And we want to understand what this integrin behaves in the defined mechanical stimulations. And this is a combination of my PhD development versus a postdoc development. So I developed this, we call live cell dynamic force spectroscopy method, or biomembrane force probe. In this system, we grab a single platelet, and we apply the mechanical force to these platelets, and then to probe integrin behaviors on the platelets. And the red blood cells serve as the spring constant to allow us to probe the mechanical property of the integrins at the different conformations. So with the first two experiments we did, we used two different ligands, fibronectin versus fibrinogens, and to probe integrin behaviors of the platelets. Two conditions: one is untreated, one is the agonist ADP stimulated. And as we expected, we see the integrin binds with higher affinities and longer lifetime. And then to mimic the mechanical stimulations from the blood shear, so we developed this, we call it dual force biomembrane force probes. So we apply the mechanical stimulations through the probe 1, and then we switch to the probe 2 and to probe the integrin conformation or integrin affinity with either ligands or conformation-specific reporter antibodies. And what we find first is, in response to mechanical stimulations, these platelet integrins actually binds intermediately relative to untreated versus agonist-stimulated conditions. Lifetime shows the same. And then we apply the same conformation reporter antibodies as we've shown at the beginning. We apply a series of conformations recognized antibody. What we found is, in response to mechanical stimulations, integrin actually binds to half of these antibodies recognized, called extended close conformations, whereas the agonist stimulated platelets can bind to all of these antibodies. Indicates the integrins on those platelets are fully activated. So. And this is another mechanical analysis we developed on top of this for spectroscopy assay. We can probe integrin stiffness at different conditions. And what we show here is with the mechanical force stimulated platelets, actually it consists of a mixture of low affinity integrin versus intermediate affinity integrin, whereas the agonist stimulated platelets, the integrin population has 3 colonies and with a relatively different percentage. So because the limit of time, I don't have enough time to show all the data, but I want to give you a summary of this work. Is we actually mapped the integrin conformation and activation landscape for 2 pathways. One is biomechanical pathway, we claim is the new one, and the classic biochemical pathway. What we show here is for the biomechanical platelet integrin activation pathway, there is intermediate transient state, and the integrin can transient to that state and then wait for the next stimulation to fully mature it. Looking forward, why this is important, two things about very important. One is this, it's of the great significance to tissue engineering, and the second, I think, is to inspire the new integrin target therapeutics, because there's lots of money has been invested to develop the integrin therapeutics. And as this diagram summarized from the Morphic Therapeutics company, the available integrin drugs are mostly antagonists that binds to extracellular domains, and these inhibitors or antagonists usually strongly, completely block the integrin functions, and that can cause side effects. In the thrombosis context, most of current integrin antagonists cause the severe bleedings. And what we believe is this new intermediate state
15 seconds
versus the full state can show us a new balance point that I can target the intermediate states, only the full states without targeting the intermediate state. Then we can reduce or mitigate the bleeding side effects of the integrin antithrombotics. I want to thank for my PhD labs in Georgia Tech, Professor Qing Zhu. My postdoc labs led by Sean Jackson, and also my supervisor, Simone Yu Ping, collaborators from UTS, Chen and Dayun, and also other collaborators from North America and China. Also, this is the last slide. This is a beautiful Australian map-like platelets images taken by the super resolution microscopy developed by my collaborators sit behind Dr. Peter Chen. We define all the dots that you shown here is the integrin distributions on a platelet, and we using this imaging technique, we'll continue to understand how these integrins